Hello everyone. Last time we are discussing about elimination in biopharmaceutics. So today's session we are going to discuss about bioavailability and bioequivalence. So my name is SPN Kumar, and today we are going to discuss about bioavailability and bioequivalence. So what is the necessity of determining this bioavailability and bioequivalence? So we are going to see all the aspects related to these two things. So let us first define what is meant by bioavailability. It is defined as rate and extent of absorption of unchanged form of the drug into the systemic circulation. So whenever we are taking a drug, the drug in its unchanged form, suppose if we are taking any paracetamol, before it is getting metabolized into its metabolites, the unchanged form which is entering into the circulation, the rate and amount, for example, it is entering at a rate of 10 mg per ml, and after 1 hour we are seeing at least 20 mg of drug in the circulation. That is its bioavailability. Not every drug is having 100% bioavailability. Some drugs they may have lesser bioavailability, like 30% or 40%. So that means if you give 100 mg of drug, only 30 mg will be available in the circulation. So this leads to problem. So we need to give additional doses. We need to increase the frequency of dosage such that the patient receives 100 mg of the drug in his systemic circulation. So bioavailability is the actual or fruitful amount of drug present in the systemic circulation. So bioavailability of a drug depends mainly on three important factors. So what are they? Pharmaceutical factors. So these are formulation related factors. How are we going to design, or how we are going to manufacture our tablets? All the formulation related aspects will be studied under these pharmaceutical factors. Then patient related factors. So even though we are taking all the utmost care to manufacture our tablet, there is some patient to patient variation. This variation may be inter-subject variation or intra-subject variation. So these things can cause differences in the bioavailability. One patient will have more therapeutic response, the other patient will have lesser therapeutic response. So this is called as variability, and it differs from intra-subject to inter-subject variability. Next is route of administration. Suppose we are giving a tablet in a parenteral form, so obviously the tablet is going to reach its 100% bioavailability. So that's why the dosage which is given through parenteral route, especially IV, it is called as absolute bioavailability, which means there is 100% amount of drug directly flushed into the systemic circulation. So what are the objectives? So why we need to learn about this bioavailability? Main thing is for the development of new formulation. So we already have a injection of a given medicament API. So I want to make this API, active pharmaceutical ingredient, into a tablet or into a gel or syrup or a capsule. So we need to study what is its bioavailability. Is it same as that of injection, or obviously it will be lesser? So it should be in a permissible limit. If it is having too little bioavailability, then we can't go for such a dosage form. We will take alternative dosage form to increase its bioavailability. So it also helps in determination of influence of excipients, patient related factors, and possible interaction with other drugs on the efficiency of absorption. So it not only tells me how much amount of drug that is entering into the systemic circulation, it also gives info about the influence of excipients. Sometimes we may incorporate more super disintegrants, or we may incorporate permeation enhancers such that super disintegrants, or we may have super disintegrants, or we have this permeation enhancers to have more drug release into the systemic circulation. So control of quality of drug product. So once you develop the drug product, is it 100% bioavailable? If not, how much of it is bioavailable? Is it high or is it low? If it is low, we've had to manipulate some physicochemical parameters so as to increase the bioavailability. Sometimes your bioavailability may be limited by solubility, or sometimes the bioavailability will be limited by permeability. That's why accordingly the drugs are classified into 4 classes according to BCS classification. So those drugs which are having lipophilic nature, those have a rate determining step with respect to solubility. Those which are hydrophilic in nature, those have a rate determining step with respect to permeability. So it also helps in the development stages of the drug. In the primary stages, we can rule out whether this candidate is an ideal candidate or it's not suitable for my formulation by using this bioavailability parameters. So we can divide our bioavailability into 2 things. One is absolute bioavailability, the other one is relative bioavailability. So any comparison with respect to IV. Why? Because IV will have 100% drug release into the systemic circulation. So hence, if you compare any drug with respect to this IV, this is an absolute bioavailability. So we are comparing our marks with respect to this 100% marks in systemic circulation, which is achieved by IV drugs, intravenous drugs, and we are also correlating this AUC with dose values of same IV and oral, but this time, since it is a division, we have to give a crisscross manner. So if AUC takes oral in the numerator, dose will be taking IV in the numerator. So this will give you an absolute bioavailability because we are comparing it to 100% circulation. But imagine you are comparing 2 drugs, each of them is a tablet. So there is a tablet which is already well nurtured in the market. Now we are going to compare this tablet with a newly formulated tablet with a other set of excipients. So this is having a different set of excipients when compared to the already popular drug in the market. Now we can compare. So you can compare one tablet with the other tablet. So there is a test drug. So this is a test tablet or a standard tablet, and we are comparing with that of a standard tablet. So this is standard, and we are comparing our test tablet with this of standard tablet. So such a comparison of AUC between test and standard is called as a relative bioavailability, because here neither the standard is 100% nor the test is 100%. So this comparison is not with respect to 100. This comparison is somewhat lesser than 100. Hence it is called as relative bioavailability. So absolute bioavailability is in comparison with IV. Relative bioavailability is in comparison with some other formulation whose bioavailability is less than 100%. So next, how can you assess this bioavailability? Now you understood what is bioavailability and why it is important. It is used for the development of dosage forms, etc. Now how can we estimate this bioavailability? So there are 2 main models. One is pharmacokinetic models, the other is pharmacodynamic models. So what are pharmacokinetic? So plasma level time studies. Suppose we are plotting a graph between time and plasma concentration of the drug. So we are plotting a curve, so that will give you a plasma level time study. Now same thing, if you are taking time and you are measuring the amount of drug which is excreted in the urine per a given time interval, it is a urinary excretion study. So other than that, we can also have pharmacodynamic methods like acute pharmacological responses or therapeutic responses. So we will discuss about each and every side heading, every header in the coming slides. So first one is plasma level time studies. So under plasma level time studies, we are going to measure how much amount of drug is present in the plasma with respect to the time. So for this we are going to take 3 parameters into consideration. So just remember CAT, C-A-T. So Cmax, Tmax, this is concentration maximum, time at which maximum concentration occurs, and the amount of drug release. So remember CAT. So that's what we will see in the next slide. So under plasma level time studies, we are going to see the Cmax, peak plasma concentration, Tmax, time at which peak plasma concentration occurs, A is amount of drug or area under the curve, which will give you a direct measure of amount of drug in the plasma. So plasma drug concentration versus time profile. This is a typical illustration of how a plasma level time curve will be looking. So we are plotting a graph between time and plasma concentration of the drug. So you know that there are two zones. One is the minimum effective concentration. Your drug should always be greater, your drug concentration should always be greater than MEC and should always be lesser than the MSC, maximal safe concentration, because if it is beyond this it will result in adverse effects. If it is below this it will result in therapeutic failure. So if you are measuring the amount of drug under this curve, that is called as area under the curve, whereas the peak point at which the concentration is maximum in the plasma, if you reach to the y-axis, since y-axis is a C term, so this is called as Cmax, and since the x-axis is a T term, this when it is interpolated to here onto the x-axis, this is called as peak time at which maximum concentration of drug is present in the plasma. Next. So here is a description about Cmax and Tmax. Let us see the definition. So maximum concentration of the drug obtained after the administration of single dose. Once after you administer the drug, the maximum concentration that is appearing in the plasma concentration versus time profile, it is called as Cmax, whereas the time required to achieve this maximum peak plasma concentration is called as Tmax. So both of them are very much important to calculate the bioavailability. Higher these two values, higher will be the bioavailability. So next is AUC. So AUC is the measurement of extent of drug bioavailability. So AUC is the measurement of extent of bioavailability. So let us see what is the formula of this AUC. So AUC is a product of plasma concentration with respect to the differences in time interval. So how can you measure this AUC? So AUC can be measured by plotting this C1 plus C2 into T2 minus T1. So let us take the, suppose there are two concentrations, C1 plus C2 into T2 minus T1. So the differences between these time intervals T2 minus T1 plus these two plasma concentrations will be giving me the area beneath this segment. So if you sum up all these things, sum of C1, C2, C3, C4, so on, you will be getting the area under curve. So you will be getting area under the curve. So this is called as trapezoidal rule. Since it is looking like a trapezium, it is called as a trapezoidal rule. So cut and weight method, other than trapezoidal rule, graphical estimation, we can also do one more thing. So a sample curve, a calibrated curve can be taken. It will be broken into small pieces, and each weight in the sense not the actual experimental weight, the frequency of that particular polygon, if it is taken and measured statistically, it can also give your AUC value. So we are also having one mechanical method that is called a planimeter, by using which we can plot the curve. So this one is fixed onto a paper. We can, depending on the area, we can plot the area under curve. So this one is having a drawback of having high chances of human error during operation or during this handling. So counting the squares, otherwise in the graph, if you are plotting in a normal Cartesian plate, you can count the number of squares. Since each square is 1 centimeter square, we can count the total number of squares to get the AUC value. So that is counting the square method, or so this is how the normal graph will be looking. So we can count this number of squares. So this one is 1 mm and this one is 1 centimeter square. So if you are counting the total number of squares under the curve, you are going to get the AUC value. So by using any of these methods, a trapezoidal method or a planimeter method, cut and weight method, or this counting the square of methods, we will get the value of AUC. So Cmax is done, Tmax is done, and AUC is also studied. Now how we can determine the bioavailability for a single dose as well as multiple dose. See, some drugs, they will show their effect with a single dose, but for chronic diseases we need to take multiple dose in a given day. So then your formula has to be manipulated to incorporate that factor. So if it is taken with a single dose, we'll just take AUC and multiply with the dose, and if it is a multiple dose study, we are going to multiply AUC, dose, and there is this tau. Tau is frequency or the dosing interval, frequency with which a tablet is given, or dosing interval. Say for example, a tablet is given 2 times a day, its dosing interval tau value is 2. So we are going to multiply 2. So the point is, each time the drug is given, it has to be multiplied into 2. because here tau is 2, so which will give me a relative measure how the drug is behaving in the body in the systemic circulation. Next, urinary excretion studies. So whatever we have done until now, it is with respect to plasma. So a drug when disappears in the GIT and appears in the systemic circulation, we are going to measure it. Similarly, this is giving me a measure of absorption. Similarly, we can also do one more thing. Suppose the drug, when it disappears from circulation and appears in the urine, which is also directly proportional to its bioavailability. So higher the amount of appearance of drug in the urine, higher is its absorption profile. So this is with respect to excretion. So AUC can be studied either through absorption as a criteria or excretion as a criteria. So this studies excretion as a criteria. So here we are so previous method, the problem is it is an invasive method. We need to take a blood sample from healthy volunteer, which is not approved all the time. So hence this non-invasive method can be used. So urine can be collected from the healthy volunteers. There is no need of informed consent, since urine is a non-invasive method of collection of sample. So we are going to take time versus amount of drug which is excreted per every time interval, 5 minutes or 10 minutes. So here also we are going to get 3 parameters, just similar to Cmax. In plasma level studies, we'll get Cmax, which here is called as DXUB-D data, which is the rate of excretion of drug in the urine. Next is just like Tmax in plasma level concentration, here we are going to get Tumax, and similar to AUC in the plasma level curve, we are going to calculate XU infinity. So there is the discussion about these terms. So dxu by dt max, it is the value which is giving me the extent of absorption and which is indirectly giving me the rate of excretion. So dxu by dt is actually the rate of drug which is excreted from the urine, which is a direct measure of absorption. Higher is the absorption, higher will be the excretion. So Tmax is the maximum time at which the maximum excretion occurs, whereas XU infinity is the cumulative amount, similar to AUC in the plasma drug profile. So here also just like AUC, so instead of AUC we will be writing XU infinity. So this will be compared with IV in single dose study, and this, since it is IV, we can call this bioavailability as absolute bioavailability. Since it is test versus standard, this is called as relative bioavailability. So if it is a multiple dose study, same formula which has to be multiplied with tau. Tau is dosing interval, how many times it is given in a given day. So these two previous methods, plasma level concentration or urinary excretion data, that is pharmacokinetic model. How can you estimate these drugs in pharmacodynamic models? We can use acute pharmacological response. That means suppose our drug, if it is having an acute response, suppose it can change the EEG value or it can change the ECG value, so that is an acute response. So such a acute response can be identified by using this acute pharmacological response method. So not every drug can produce this acute response, only some drugs can produce acute response. So that is main problem is it tends to be variable, patient to patient variability will be present, and it may occur if there is some active metabolites. Some drugs like risperidone or psychological agents, they will have active metabolites which will result in more therapeutic response than in the initial stages. For such things, this may not be applicable. So therapeutic response method. So apart from acute pharmacological response, we also have therapeutic response. Suppose your drug is used in a chronic condition like BP or epilepsy, we can use this therapeutic response method. The major drawback is quantitation is not applied for all drugs, so some responses cannot be quantified. Say for example BP, we can control the BP only up to a certain level, that is until the blood pressure reaches 120/80. Now if you are even giving higher doses, your blood pressure won't go beyond this 120/80 because our homeostatic mechanisms are going to protect our standard value of blood pressure. So quantification is not possible. That's the main issue with the therapeutic response models. So what are the clinical observations? So in the clinical trials, we will estimate this bioavailability and bioequivalence to see whether your drug can be approved or cannot be approved, whether to go with a drug or not go with a particular formulation of the drug. So we can also use this in vitro studies. So we will compare these results from the in vitro and we'll take another stand with in vivo studies. So if both of them are having correlation, in vitro in vivo correlation, then a drug will be successfully approved or released into the market. So apart from bioavailability, in this third chapter we also have bioequivalence. So what is bioequivalence? So you are giving a tablet and you are giving a capsule. Both of them are consisting of paracetamol and both of them are releasing 100 mg of drug into your circulation. Then these two formulations are said to be bioequivalent. Bioequivalent in the sense, no matter what is its physical form, they are going to release same amount of drug into the systemic circulation. Then it is called as bioequivalence. So what is the objective of using bioequivalence? So it is done to sure that your substitute, suppose sometimes without going for clinical trials again, if both of them are bioequivalent, we can easily approve a drug. So previously a tablet is present for paracetamol, now a capsule is also showing same release, so then it can be easily released into the market without having trials from the beginning of your clinical trial development. So it can also be used for equivalence studies. So this equivalence can be chemical equivalence or bioequivalence. If the chemical component of both the formulations are same, it is chemical equivalence, and if its biological performance from both the formulations is the same, it is called as bioequivalence. So pharmaceutical equivalence, which means, so whatever your dosage form, different dosage form like tablet or capsule are there, if both of them are performing with the same amount of bioavailability, it is called as bioequivalence. So what is the bioequivalence criteria? The criteria is they have the same disintegration time and same dissolution rates, but they have different shape. Like a tablet may be different and a capsule may be different, or the release mechanism may be different. Sometimes they may be released either by diffusion or sometimes they may be released by erosion. The release mechanism is different, but end of the day both of them are releasing same quantity of the drug. So their packaging may be different or their excipients. So nature of excipients used here, we may use some other colors, flavors or preservatives, whereas the capsule may have different set of excipients, but finally both of them are going to release same amount of drug. So these bioequivalence studies are helpful to find out the pharmaceutical substitution compounds or alternative dosage forms. So therapeutic equivalence in the sense, even though the API is different, if they are producing the same response, same level of response, then it is called as a therapeutic equivalent. Suppose adrenaline tartrate may have different effect with respect to plain adrenaline. If both of them are showing same responses, it is called as a therapeutic equivalent. Caffeine citrate or caffeine editate also come under this therapeutic alternative. So therapeutic substitution, if ampicillin is used instead of amoxicillin or ibuprofen is used instead of naproxen, which means both of them are NSAIDs, but they're chemically different, completely different, but if they produce same amount of response or same amount of action, it is called as therapeutic substitution. So that concludes what is meant by bioavailability and what is meant by bioequivalence. So these are very much essential in the clinical trial development and in the development of new dosage forms. In the next class we are going to come up with a new topic. So that's it for today. Thank you.